This podcast is part of a special series highlighting work to be discussed at the 2024 Cancer Neuroscience Symposium, which is hosted by MD Anderson Cancer Center and Wiley on February 28th through March 1st, 2024, with options to attend virtually or in person. Learn more and register at mdanderson.org/Cancer Neurosymposium.
Hello everyone, and welcome back to Beyond Cancer. Today I'm here with Dr. Varun Amit and Dr. Bernie Matson, and we are interviewing Dr. Varun Venkataramani as part of our 2024 Cancer Neuroscience Symposium series.
Hey, thank you, Sahana, and thank you so much, Dr. Venkataramani, for joining us today. I am really honored and privileged to have you here on this podcast. So, Dr. Varun Venkataramani is an MD PhD faculty at the University Hospital Heidelberg, also a group leader at DKFZ, which is the German National Cancer Institute. His lab is focused on gliomas and glioblastomas and their interaction with the nervous system, looking straightforward at neuroregulation of cancer at the CNS level. Varun had multiple high-impact publications, Cell, Nature, and he's doing fascinating research for the first time, looking at those direct interactions and also how those neurons at the CNS synapse with the cancer cells. Varun, thank you for joining us today. I had a great time, meeting that you held in Heidelberg, and to hear you presenting your data, your group's data. And can you tell us a little bit about you, your career path, and your lab?
Yeah, thank you, Varun, and thank you everyone for the invitation. Yeah, so I'm a neuroscientist, I'm a neuro-oncologist, and I am interested in understanding how glioblastoma and tumor networks are organized. So a couple of years back, we found that tumors can not only communicate with each other via gap junctions, but also that neurons can communicate with tumor cells through bona fide synaptic contacts. And I'm interested in understanding these different kinds of multicellular tumor networks, how they influence tumor biology, treatment resistance, and how we can use disconnecting therapies as a novel approach to treat these incurable brain tumors.
Thank you for that. And, just to give our listeners a brief overview and introduction to what brought you into cancer neuroscience, I wanted to ask, what do you find most intriguing about the field, and why did you begin that? Why did you decide to pursue that?
Yeah, so our path to cancer neuroscience was more of an accidental one. So we were a few years back, from Winkler's group found that these tumor cells show neurite-like extensions, they call them the tumor microtubes, and he approached us and wanted to understand how they look like in electron microscopy, so to understand the ultrastructure of these tumor microtubes. And basically, when we did these analyses, by accident we found that indeed there are not only these tumor microtubes, but there are also synapses of the tumor microenvironment that connect to these tumor microtubes. So this was basically intriguing for us, and what we wanted to understand more is whether this is how this basically contributes to brain tumor pathophysiology. And I think we found that basically this is one of the hallmarks of glioma growth.
And kind of following up on the hot publications that you've had, both in, at least the ones I'm familiar with, in Cell, broadly speaking, do you have any views or opinions that really challenges the current dogma or current train of thought for the community and field of cancer neuroscience?
So I think it's a rather new field, so I'm not sure whether there's anything, whether there even exists something like things that again go against dogma, I think.
My take on that from your answer is that there's a lot of opportunities within the field of cancer neuroscience to make an impact.
Yes. So,
is there anything that really is exciting you the most to jump into those gaps of knowledge that exist?
Yeah, so I think my take here is that I think we have to, basically I want to bridge three major challenges. I think on one hand, I think we need to do much more on the basic science side of cancer neuroscience, to really understand what kind of components play a role in these tumors, both inside and outside of the brain. What is the anatomy of these tumor networks? How are they molecularly composed? What is the physiology? I think the second huge challenge, I think, will be also to develop appropriate methodologies to study these phenomena and to really have tailored methodologies to study cancer neuroscience. Again, both inside and outside the brain. I think the goal that we want to achieve in the end is to really come up with novel therapeutic approaches, to come up with neuroscience-instructed cancer therapies to really make an impact also on patient outcomes. I think these are the three challenges that I want to tackle in the next years.
That's awesome, Varun. You mentioned briefly, the extracranial tumors, and we've shown that in extracranial tumors, this cancer has the capacity to hijack the nervous system and actually utilize it, quote unquote, for its own purposes. Can you briefly tell us a little bit on how glioblastoma hack the global mechanisms in brain that pre-existed physiological neuronal activity in order to promote tumor growth or resistance to therapy?
Yeah, that's a great question. I think we are basically only at the beginning of understanding the impact of neuronal activity, and I think there are many challenges still ahead and many questions still open. But to briefly summarize this, so I think on the one hand, there are very basic neurodevelopmental pathways are hijacked, and we've been recently studying also how this has been hijacked in glioblastoma invasion. And what we basically see is that there are neuronal-like cell states, neuronal precursor-like cell states, and also oligodendrocyte precursor-like cell states that are hijacked for invading into the brain. So basically, that certain cell states, molecular cell states, are responsible for invading the brain. We do see this as well on a phenotypic level, on the second layer, that basically mechanisms of invasion are hijacked that are normally used by immature neurons that wander through the brain during brain development. And in the end, what we do see is also these direct connections between neurons and tumor cells, synaptic contacts on tumor cells that mediate both invasion and proliferation of tumor cells, mainly with all three layers. And I think there are many questions open. For example, also what is the role of neuronal activity and also the synapses, for example, in therapeutic resistance. I think this is something we don't yet know, but what is definitely an important research area in the future.
Yeah, so you mentioned the neuronal activity, and one thing that comes to mind, for example, if you see an increased activity that might be associated with more aggressive behavior of the tumor. So, we have drugs to target it from other worlds, like, you're a neurologist, we have epilepsy drugs, anticonvulsants, those kind of things to keep the electrical activity kind of like inhibited. Do you see those drugs being repurposed down the road for cancer treatment?
Yeah, also very good question. I think that's something exactly what we're currently thinking about, is to repurpose at least certain anti-epileptic drugs with certain mechanisms such as upper receptor antagonists, such as the FDA-approved antiepileptic drug perampanel, where we see, at least in preclinical models, that this is indeed in fact a cytostatic effect on glioma growth and also an effect on glioma cell invasion. I think that's, again, a challenge of cancer neuroscience and the challenge of basic neuronal and neurodevelopmental mechanisms. Hijack is to understand whether there's a large enough therapeutic window of tackling and making use of these mechanisms while still preserving the integrity of the central nervous system. I think this is something that we need to explore in the next few years, and also think about drugs that have a large therapeutic window that do not necessarily affect the function of the healthy central nervous system, but only is more specific to brain tumor cells.
Well, speaking of you mentioned AMPA receptors and drug repurposing as a therapeutic strategy. That brought to mind the idea of neuroplasticity. And so could you speculate on how the nervous system can remodel the tumor microenvironment to really, I guess, open up as a therapeutic window to enhance the efficacy of these cancer therapies?
Yeah, I think the idea of also not only, I think there is certainly a lot of bidirectional communication between the nervous system, neurons, and tumor cells, and I think also that if we especially think about gliomas, there's always also we get more and more data from various publications in the last years that there might be indeed such a vicious cycle of neuronal hyperexcitability, so basically epileptic or epileptiform neuronal activity, and glioma growth, and in the end, glioma growth again also affecting neuronal hyperactivity and also certainly neuron plasticity for that matter. And I think, again, also when we think about more basic mechanisms of neuroplasticity, also these kind of mechanisms have been recently described, for example, by Michel Monchi's group. So I think we are only at the beginning of understanding how these bidirectional mechanisms really work, and I think that is something that we really need to analyze and measure also in more detail to get a fuller picture how neuronal activity and what kind of neuronal activity also really drives the plasticity of tumor cells and vice versa, which neurons are really involved. Are all neurons involved to a certain degree? There's only a subset of populations, and how again are neurons manipulated by tumor cells, and how can neurons then again, and which kind of activity, so which kind of activity patterns really modulating and also tumor growth.
So Varun, what do you think are the hurdles or the biggest roadblocks that blocks us from translating those beautiful findings into the clinic?
Yeah, I think there are probably a lot of challenges yet ahead, but I think one major challenge that I see, at least for clinical translation, is to really adapt and develop also cancer neuroscience specific biomarkers, and to really define what is a successful phase 1 and 2 trial. And I think that is something that we really need to do in depth as a field altogether, because I'm not necessarily sure whether also simple repurposing will in the end be enough to really make a huge difference. But I really see this only as a starting point to see what really can work in small cohorts of patients, and then really have targets for further drug development then that really can make a huge difference for patients.
Thank you. And then bringing it back to the symposium, I just wanted to ask you what are you the most excited about for the upcoming Cancer Neuroscience Symposium?
Yeah, I think it's a great speaker lineup. I think what's very interesting for me also, someone who is very interested in central nervous system malignancies, is to also get to understand better what also happens in the periphery, how basically these both rather distinct fields still can learn from each other, and again how what novel aspects of brain tumor cancer neuroscience will be able to learn.
You know, we just had the meeting in Germany, and this one's going to be held 6 months after, or a little more than that. It will be interesting to see, what new data people will present. In Germany, I learned that there's a lot of enthusiasm, a lot of people that are actually involved in the field, more than I thought. And I'm really curious to hear, what's going on in your group. Are you going to present any new data? Can you give us a glimpse into it?
Sure. So I'm not sure yet what we will present. So I think as you said, it's still 6 months, right? So we will certainly show something new. And I think we're working on a couple of interesting projects. And I think we are currently mostly focused on really also developing methodologies that we then in the end can apply also to understand novel biology. So I'm certainly going to present a few new technologies that will hopefully the whole cancer neuroscience field can benefit from.
That's exciting. Thank you. And to our listeners, we will see you guys next time. And thank you so much, Dr. Venkataramani, for being here with us today. And thank you, Dr. Madsen and Amit, for co-hosting with me. Bye, everyone.
This podcast is part of a special series highlighting work to be discussed at the 2024 Cancer Neuroscience Symposium, which is hosted by MD Anderson Cancer Center and Wiley on February 28th through March 1st, 2024, with options to attend virtually or in person. Learn more and register at mdanderson.org/Cancer Neurosymposium.